Sexual reproduction in flowering plants. What did we begin with? We began with the study of androecium, study of stamen. We studied the structure of anther wall. If you remember that 5 marks, very important diagram, structure of a mature anther wall or a young anther wall. What are the anther walls made up of? Epidermis, then the endothelium, middle layers, tapetum, and sporogenous tissues in an young anther. In a mature anther, instead of sporogenous tissue, you have to draw the pollen grains. I have told you that this is almost like a butterfly diagram. All the 4 tetrasporangium you have to fill it up during examination. You can't just write a large portion as they have done in your book. You have to draw the complete cellular details in all of them. At the center of this butterfly-like diagram, what do you notice at the center? Connective tissue. That you should remember. They have tetrasporangiate. Sporogenous tissues are of 4 chambers. Tetrasporangiate. Okay, all that you have to draw, and that diagram is very important. Then a mature pollen grain we discussed about. They have an outer exine wall and an inner intine. Exine is made up of a chemical substance called as sporopollenin, which prevents the dehydration of the pollen grain, which also prevents the activity of chemicals on the pollen grain. That's the reason pollen grains can survive for a long duration of time. So then we discussed about the maturation of pollen grain or formation of a 2-celled or 3-celled pollen grain. Yes, steps ittadu. Microsporogenesis, microgametogenesis. In textbook alli microgametogenesis badlu avaru em kottidare formation of a mature pollen grain. That is nothing but microgametogenesis. What is microsporogenesis? One of the functional microspore mother cells, they undergo reduction cell division to form 4 microspores, which are all held together as a tetrad. So the microspore gets separated from the tetrad by the dissolution of the callose plugs, which are holding them together, callose walls. So the microspore, they then develop to form what? The microspore develops, they undergo mitotic cell division, unequal cell division to form a larger vegetative cell and a generative cell. The generative cell separates from the wall and floats within the vegetative cell. So a cell within a cell. If the pollen grain is released at this stage, we call it as 2-celled pollen grain. So nearly 60% of plants release the pollen grain as 2-celled pollen grain. 40% of plants, they release the pollen grain as 3-celled pollen grain. 3-celled pollen grain that generative cell undergoes one more cell division, mitotic division it is, to form 2 sperm cells. So there are cells within a cell. Cells within a cell you notice in 3-celled pollen grain. So 40% of plants, they liberate the pollen grain as 3-celled pollen grain, which we had discussed about. Then we studied about gynoecium, pistil andre the carpels. Then we studied about style and the stigma, where they receive the pollen grain. All that we had studied about. And again here we studied the structure of ovule. Okay, so you have studied the structure of an anatropous ovule. This is what the diagram was about. Anatropous ovule with 2 integuments, and there was an embryo sac and nucellus. Okay, this we had studied about, and this minute opening we call it as micropyle, and this as chalaza. The region opposite to it as chalaza. These 2 integuments were there. Okay, the funicle and the ovule, they were close together. Micropyle and funicle are close together. This is a anatropous ovule. Longitudinal section of anatropous ovule we had studied about. One of the cells of the nucellus, they become large and they start functioning as megaspore mother cell. So I have drawn diagrams for that also. Megaspore mother cell undergoes meiotic cell division to form meiosis 1 to form dyad, meiosis 2 to form tetrad. Of this 4 megaspores, only the one megaspore is functional, remaining they degenerate. That functional megaspore, now they undergo megagametogenesis. Megagametogenesis is mitotic cell division. Now they undergo cell division to form two cell, vegetative cell and generative cell. This two cell pollen grain concept. Three cell pollen grain they are going to form. That is what we had studied. Then we studied the process of double fertilization. Double fertilization and first step pollen grain germination. Then the pollen grain, before that we had also studied about pollen pistil interaction. Isn't it? So wherein the receptivity of the stigma and the viability of the pollen grain, right type of pollen grain should fall on the stigma and there should be receptivity of the stigma. Only then they can undergo germination. Pollen grain undergoes germination to produce pollen tube. That pollen tube will be having pollen contents which can be the vegetative cell and the vegetative cytoplasm and generative cell. Two cell pollen grain, every generative cell divides to form two sperm cells. Three cells are already ready-made. So the entry of pollen tube into style, entry of pollen tube into ovary, entry of pollen tube into ovule. The entry of pollen tube into ovule, it can be of three methods. If it enters through micropyle, we call it as sporogamy. If it enters through chalazal region, the pollen tube, we call it as chalazogamy. If the pollen tube enters through integument, we call it as mesogamy. Whatever the pathway of entry, they ultimately should reach the micropylar region and the contents are deposited in one of the synergids where the filiform apparatus receives the pollen tube contents. Another synergid degenerates. So they would be having two sperm cells. The sperm cell which is rich in plastids, they fuse with the egg cell to form the zygote. The sperm cell which is rich in mitochondria, they fuse with the secondary nucleus to form primary endosperm nucleus or primary endosperm cell, PEC. So this is what is about double fertilization equals syngamy plus triple fusion. The process of fusion of male gamete with female gamete, we call it as syngamy. The process of fusion of sperm cell with the secondary nucleus, moor fusion ಆಗುತ್ತೆ ಅಲ್ಲಿ. ಎರಡು polar nuclear fuse ಆಗುತ್ತೆ, ಒಂದು sperm cell fuse ಆಗುತ್ತೆ. ಅದಕ್ಕೆ ಏನಂತೀವಿ? Triple fusion ಅಂತೀವಿ. What do you call it as? Triple fusion. Double fertilization equals syngamy plus triple fusion. Post fertilization, what are the changes? The zygote develops to form the embryo. The primary endosperm nucleus develops to form the endosperm. Okay, so that is what we had discussed. Then we also discussed the formation of a dicot embryo and a monocot embryo in the previous class. And today's class we completed with the apomixis and polyembryo. These are all the concepts which I am putting it in a nutshell for you, the entire chapter that you have to remember about. Important diagrams, mature pollen grain, not mature pollen grain, transfer section of anther wall of mature anther wall or young anther wall. They can also give you the diagram of LS of ovule, anatropous ovule. That also they can give you the diagram as. Then they can ask you the megasporogenesis, development of embryo sac, or they might ask you to draw the diagram of embryo sac. Only once they have asked the diagram of the fertilization process, where you have to show the pollen grain fertilization. You will end up noticing that only once they have asked for that diagram to be drawn. Okay, so be aware about what diagrams you have to practice and the concepts. Important take-homes are double fertilization is unique in angiosperms, dicot embryo is heart-shaped, monocot embryo where we call the cotyledon as scutellum. Okay, then finally apomixis and polyembryony, how it helps in stabilizing the hybrids. And we can have the same copies of hybrids by employing apomixis. Okay, so these are the things that we should remember about the entire chapter. That completes our sexual reproduction in flowering plants. Okay.